visitor this afternoon is Dr. Peter Lindeman from Edinburgh University, and I'm pleased to welcome the Erie County Community College students here today. Welcome. Peter.
Thank you. Well, as of the start of this semester, I didn't have a talk of my own to give for this symposium, but back in September, the last 2 weekends of September, I got away for a couple of long weekends to Illinois and did some turtle survey work, and at the end of it I realized suddenly I had a talk that I could give. So I want to talk about what I did in Illinois and the concept of Linnaean and Wallacean deficits in biology, especially the Wallacean deficit. The Linnaean deficit is something that people have been talking about in recent years as our incomplete knowledge of all of the diversity of life on Earth. So every year we get something like 6,000, 6,500 new species described, and we get some of those sunk as synonyms later on. So our understanding of the world's diversity is always in a little bit of taxonomic flux. And then the Wallacean deficit, once you stabilize on how many species you're going to recognize, there's the question for each species, where exactly does it occur? How wide-ranging is it? And so if you know all the species you have, and if you know their range, I've spent a fair amount of my career working on especially the Wallacean deficit. I work with map turtles and sawbacks of the genus Graptomes, and back 9 years ago when I put out my book on map turtles and sawbacks, one of the tables in the last chapter of the book lists all the new tributaries recorded for the 14 species of Graptomes. That was from 1995 till 2013 when the book was published. And for somebody like me, 1995 still seems pretty recent. So recent range extensions for all these species. So a lot of these are my own, and then a lot of them are attributable to various colleagues who have worked with map turtles and sawbacks. In addition to these, in some of the known tributaries of river systems where these turtles live, we've also established numerous upstream range extensions, sometimes very lengthy upstream range extensions where we find a turtle 50 or 100 kilometers up above where anybody's ever recorded it. So there's a bit of a Wallacean deficit with regard to these animals for sure. So I spent 2 weekends in September over in southeastern and south central Illinois. This is the Little Wabash River. You can see it travels through many counties of southeastern Illinois, and as far as I have been able to determine with various searchable databases and my own databases I've put together on the map turtles, but searchable databases for other turtles as well, I can't find any records of any turtles ever collected anywhere in this Little Wabash River, which is not such a little river. And then the next to it, just north of it, is the Ambaraw River. That's a French word. Looks like embarrass, the Embarrass River, but it's, you'll only embarrass yourself if you go to Illinois and call it that. It's the Ambaraw, which is from a French word which can mean embarrassment, but it also means difficulty or entanglement, and it's a reference to all the deadwood in the river. So I think if they had named it in English, they would have just called it the Snag River. And I also looked on Bumpus Creek and a little bit up on the Vermilion River, all tributaries of the Wabash River, which has not had a lot of map turtle or other turtle work done throughout it. Now, I call this area the Corn Wilderness because I was really struck on these 2 weekends by all the times that I would drive from one bridge to another to look at the turtles there and take photographs of them. and I might be driving 30 or 45 minutes and hardly see another person or another vehicle on the road. Or I'd walk out onto a bridge to look for turtles, and I'd be out there for 10 or 15 minutes and hardly see a vehicle, or maybe not even see a vehicle pass over the bridge in that time. Several of these counties, there's 8 or 10 contiguous counties which literally don't have a single human being living in them. Okay, not literally, but it just seems like that if you're spending time there. Cornfield after cornfield after cornfield, but some of the county populations, most of them in this area are below 20,000 for the whole county. Several of them are below 10,000 for the county. So that's why I'm calling it the corn wilderness. Here's a look at the Little Wabash River. This is up close, getting close toward the headwaters up north, and then this is down south, getting close to where it joins the main Wabash River. Fortunately, you cannot plant corn in a riverbed, so the rivers are in pretty decent shape. They have riparian zones right along the edges of them that provide the deadwood for the turtles to bask on. So I'm going to go through the species one at a time, and I'm going to use maps taken out of the Field Guide to Amphibians and Reptiles of Illinois, published in 1999 by Phillips and Brandon and Mall. And they use purple for recent vouchered records and light blue for photographic records like what I'm providing. And then the slanted ones are pre-1980 records. So my first species is one of the map turtles, Graptomys wachatensis, and here are all the places I found them in the Ambraw River and the Little Wabash River, and it comes out to 5 new county records and then one county where they hadn't been recorded since sometime before the 1980s. And then a similar species to them is Graptomys pseudogeographica, the false map turtle. 7 new county records along the Ambraw, Little Wabash, and 1 first record up here in Cumberland County, first record since, turns out that one goes back to the mid-1940s, and they haven't been recorded. Obviously, people just not looking for them because they're fairly plentiful up and down both these rivers. So both those species of Graptomys occur in the lower Ambraw and the lower Little Wabash. So we go on to the common map turtle. This is the same species we have on Presque Isle. I found them, found common map turtles up here in the upper Ambraw, and this is part of the Vermilion River, and then that's that little Bumpus Creek that is in between the 2 other rivers. So this species in this part of Illinois prefers some of these smaller creeks, the upstream part of the Ambraw, Bumpus Creek, and Vermilion is fairly small and shallow as well. I found no overlap of them with the other 2 species. There are some other areas in Kansas and Missouri and Indiana where there's limited overlap between this species, Geographica, and the other 2, Pseudogeographica and wachatensis. I don't know, there may be some limited overlap in these river systems as well, but I wasn't able to find where they overlapped. So it was either this species or the other 2 that I was tending to find. Painted turtles, no new county records for them. They've been pretty well covered because they live in smaller, like small farm ponds and things like that, which have been better sampled throughout this region. I got 1 county where they hadn't been recorded in a long time up here, but very few new county records left for them. I only really looked in 1 place where I might have gotten them and didn't get them there. And then red-eared sliders, the world's most ubiquitous turtle species. These things have been found on every continent that has any kind of turtle because of the pet trade. They're They're kind of a low-grade invasive species. They're not really bad as invasives go, but they are successful in invading a lot of places around the world. Here, though, they're native, so it's always fun to get to talk about Trachemy scripta in their native range. They get talked about more in their non-native range than their native range. And if you can get 5 new county records for Trachemy scripta, for crying out loud, you know you're in a place that people just haven't done much prospecting for turtles. And then 2 soft shells in both in the lower Ambra River. I don't know why I didn't see these in the Little Wabash River. It looks like habitat, at least for the second species I'll show you on the next slide, the spiny soft shell. But I found both of these only in the lower Ambra here, and got 2 new county records and 1 updated record for the smooth soft shell. And then the spiny soft shell, I got 3 localities for it, but all in the same county, so 1 new county record for that species. I have to wonder if there aren't some other new county records I missed for 1 or both of these species, especially in the Little Wabash River. So 22 total new county records and several other counties that nobody had reported anything since before 1980, in some cases decades before 1980. I think these 2 river systems, the main ones I was looking at being the Little Wabash and the Ambra, I think they are contributing to a small chunk of our wallation deficit in understanding the geographic distributions of these species. Now here's a photo from one of the new county, this is Jasper County, which has fewer than 10,000 people in it. None of them apparently turtle biologists, because I got several new county records for various species out of Jasper County. And we have a Watchitaensis there, a Ouachita map turtle, another Ouachita, all the Ouachitas are looking that way. These 2 with the more crescent-shaped marking behind the eye are false map turtles, and that's a little male false map turtle there. So in this particular photo, I feel pretty confident in telling those species apart. And I'm used to that in turtle surveys done with my long lens camera and my high-powered image stabilizer binoculars that I use to initially find the turtles and identify them. I'm used to being able to look and see various species of graptomes and then the sliders and soft shells and other species and tell them apart pretty well. But these 2 were kind of messing with my head a little bit as I was doing this work back in September. So here's the range map for, whoops, what did I do? Let's see. Here's the range map for Graptomes ouachitaensis and the Wabash area over here. And then here's the range map for Graptomes pseudogeographica. Ouachitaensis is all just ouachitaensis. There aren't subspecies described for it. Pseudogeographica in the southern states down here is called the Mississippi map turtle, and they have a crescent-shaped marking behind the eye. The crescent takes over the little spot above the jaw, and then they have a little spot below the jaw, and they have a little spot on the chin. Those spots are smaller than the eye. And you can tell them apart from the ouachitaensis, which has a big wide bar and a big spot and a big spot and a big spot on the chin. You can tell them fairly readily in these southern localities. As you go up north, they're harder to tell apart. And in fact, the northern populations used to be, for a long time, considered a separate species from that Mississippi map turtle. And they used to be considered the same species as ouachitaensis. It wasn't until the 1970s that a grad student showed with careful analysis that their head markings are different. Everything else is the same about them. The shell markings and shell dimensions, identical. If you had just the shell of the animal, you'd have no idea which one it was. But the head markings up in these northern regions are slightly different. Wachita, big fat bar, big fat spot, and big fat spot. Thinner bar in Pseudogeographica, small spot, small spot, small spot. I wasn't finding the bar to be all that thin in the Wabash River area, or the tributaries of the Wabash. I wasn't finding it to be all that thin, but the spots were smaller in the Pseudogeographica, and many of them had crescents. So it looks like this area where I was prospecting for turtles here, looks like that's in the intergrade zone for these two subspecies, the Mississippi map turtle and the false map turtle. A little more false map turtle-ish maybe, but still probably intergrades. And as I was saying, I'm not used to looking at my photos after the fact and saying, now wait, is that really a Wachita, or really, really the species I thought it was? So here are some Wachitensis that I feel really confident about. Big fat spots, big fat bars behind the eye, and the big fat spots that are above and below the angle of the jaw and on the chin. This one has a spot bleeding into a nose stripe, but they usually are separate. So all four of those within the eye there, very small spot below the jaw in that animal, and a very small spot, much smaller than the eye, for that animal. So that fits what the graduate student, a guy by the name of Dick Vogt, found back in the late 1970s studying these two. He was the first one to separate them as different species. But along the way, I got some animals that I went ahead and categorized as one or the other. So I'm calling this a false map, and I'm calling these two Wachitas, but I'm feeling a little more uneasy about them. So I hadn't really thought about this going into taking these trips in September, but I came out of taking the trips in September saying, well, not only did I get some new county records, but I need to go back to Illinois and take my canoe and take some traps. Trap some of these animals, get better, closer up photos of all their head markings, and then take some genetic samples and find a collaborator to work with on looking at them, because I want to know if my readings of the head markings really do correlate to the identification of the species, and I'd also like to know, might there not be some 5 or 10 or 20% of specimens that show genetic evidence of hybridization, and maybe, and if they do show hybridization, what does that tend to look like in terms of these head markings? So, so this little short project was something that took a tiny chunk away of the Linnaean deficit, perhaps, or the volation deficit with the 22 new county records and new tributaries for these species, but also maybe there's a little bit of the Linnaean deficit in terms of understanding just how different these two species are in this particular part of their range, in the greater Wabash drainage part of their range. And ultimately, hopefully I'll be able to get some genetic samples to answer the question of whether hybridization occurs. Thank you.
Questions for Peter?
Excellent.
All right. Thank you.
Thank you.
Well, good afternoon, everybody. My name is Danielle Beck, and I am from Pennsylvania Western University at Edinburgh. Dr. Lindemann and I this past summer have been doing research on allometry and isometry in the scaling of body mass to linear shell measurements in a variety of different turtle species. Log-log transformation of a 3D measurement regress onto a 1D measurement generally results in a slope of 3.0 if it's isometric. However, if that 3.0 slope is skewed either above or below, then it would be considered allometric. Turtle body mass scales hypoallometrically to shell length with slopes less than 3.0. And this is because juvenile turtles are circular, and then when they mature, they'll generally elongate, producing a slope that's hypoallometric. And the majority of our data basically shows that the slope is between a 2.6 and a 2.8, and we have various data that supports that. And for our research, we examined whether turtle body mass would scale at a rate of 3:1 with a geometric mean of shell length, width, and height. Now, the geometric mean is simply multiplying length, width, and height and taking the cube root of it. And we use the geometric mean because any single 1D measurement may be allometric. So using the geometric mean may cancel this out. Although it may distort variable relationships, allometry works under the principle of relative variation, meaning that the multiplicative model outlining proportions would be more accurate than the additive model, And these kinds of studies would be important for reproductive allometry in turtles, which is how clutch size, mass, egg size, and length, would relate to relative body size. And since turtles elongate more in length and width, length may not impact in our research. The first one is the common snapping turtle. The second one is the Sabine map turtle. The third turtle we looked at was the Texas map turtle. And finally, our last turtle was the common map turtle. In total, we collected 473 different individuals across these 4 different species of turtles. So we collected the common snapping turtle and the common map turtle here at Presque Isle. We collected them in Misery Bay, indicated by this star, and we also collected them in Graveyard Pond, indicated by this yellow star. We were able to collect the Sabine map turtle on the Marmontel and the Calcasieu River. The Calcasieu River is indicated by this 3 right here, and then the Marmontel is this 4th river right here. And we collected these turtles in various sites within this green oval along these rivers. And lastly, we collected the Texas map turtle in Texas, hence the name, in the Fayette and Matagorda counties. And this here is the Colorado River Basin, or drainage basin, And again, we found these turtles within these green circles. So how we collected these turtles is by using fyke nets. A fyke net has a lead net indicated right here. And the lead net, hence its name, will lead either turtles or fish into the fyke net, which is 2-chambered and acts like a funnel. And it'll direct these turtles or fish into the final chamber where they will be trapped, and we will come later with the canoe and collect them out of the fyke net. Additionally, we use basking traps, and hence their name. There's a platform on top of the actual net, this platform here and here, where the turtles will actually bask on them. And we will bring up our canoe and hopefully have the map turtles jump into the net. Whether that happens or not, it's a bit variable, but that's the goal. And then once we collected the turtle, we used a variety of different measurements, including a ruler, handheld scales. And we'll collect data including their ID, sex, the date, the site where we collected them, carapace length, carapace width, carapace height, body mass, their age, if applicable, and then if they were producing any eggs. So unlike our map turtles here, where we can just easily collect them in a bucket, our snapping turtles were a little bit more difficult. For our snapping turtles, we had to hog tie them, And hog tying is simply taking the rope here and putting it in front of the snapping turtle so that they will bite it, and then ring it around and tie it up. And the reason why we do this is because, one, we don't want our fingers gone, and two, because we actually have a loop on our hog tie in which we will take one of our scales and put the hook through that loop, and we will actually weigh them that way. And so once we collected all of our data, our field data, we found the geometric mean, and as I stated earlier, that is simply multiplying the length, width, and height and taking the cube root of it. We then log transform length, width, height, body mass, and the geometric mean. And finally, we regress body mass on length, width, and height and geometric mean to find the slope and R-squared value using S+. And these are the results that we found. Our first turtle here is the common map turtle. As you can see, the length for both male and female produce slopes of less than 3.0, again indicating that it's hypoallometric. Additionally, we have slopes above that 3.0 for our width for both male and females. Height is a little bit more variable, but generally is below that 3.0 mark. However, I want to direct you to the mean especially because the males produce a mean of 3.05, which is very close to that 3.0 mark that we predicted. Additionally, females produce a result of 3.01, once again, very close to what we predicted of a 3.0 slope. And generally, our R-squared values were better for the mean than any other individual measurement with length, width, and height. Now our sample size for the males is 76, and for the females it's 205. For the Sabine map turtles, it also produced similar results, with that length slope being below that 3.0 mark, width above, height a little bit more variable with males being above 3.0 and females being below that 3.0. However, our means for the male is 3.04 and females is exactly 3.0, which is spot on. And for our sample size for the males, we have 48, and for females, our sample size was 47. Our last map turtle was the Texas map turtle, which produced somewhat similar results. And finally, the snapping turtles. Not only were they a pain in the field, they were a pain with the results that we got. Simply put, the data, the results that we got were just simply weird. As you can see, our length is above 3.0, and even the male length is very close to a 3.0, which is weird. Males is below a 3.0, females is significantly high. Height is very, very low, 3.0. And our mean for the males did actually produce a slope of 2.94, which is relatively close to 3.0. But our females did produce a very high result of 3.31. We did, however, get a sample size for the males of 24 and females 19, which is relatively a small sample size. However, our smallest sample size is 15 with the Texas map turtles, which did produce a good slope of 3.07. So it's not just the sample size that are causing this weird result. There's something clear. There's something else. So the map turtles generally follow the pattern predicted that using the geometric mean of shell length, width, and height improve the regression model fit. But generally, especially for the Texas map turtles and the common snapping turtles, we need larger sample sizes, especially for the common snapping turtles. And then for the results for the common snapping turtles, why they were so weird, we think it might be because other body parts are growing allometrically that we did not measure in our results or in our field. Firstly, it could be the head, as you can see pictured here. Secondly, it could be the tail. Although not pictured, I'm sure you can assume what a turtle tail can look like. And then finally, the limb musculature could also be growing either hyper or hypoallometrically that could have skewed the snapper data. Thank you.
questions for Danielle?
So of the species of turtles you just went over, do you think the snapping turtle kind of had that different results because the other ones seem like they're really good swimmers? Snapping turtle is definitely aquatic, but they're not a great swimmer. Do you think maybe that's what kind
For
of
sure,
plays
yeah.
into that body shape?
Yeah, as I said, the limb musculature is far different than like a normal map turtle, and we think that the limb musculature could be growing allometrically, which could have skewed our results. And like you said, they're far different than just a simple map turtle. So for sure, there's definitely something else that we need to look at and measure to see if it's growing allometrically or not.
Other questions? Thank you.
Thank you.
Our next speaker is Eva Kerr. She's from Allegheny College, and she'll be talking about edge habitats and sampling type on conserved properties in northwest Pennsylvania.
Thank you.
Salamander.
Hi, and yeah, I worked with Dr. Pierce and Dr. Bradshaw Wilson on my senior project at Allegheny College, and I worked with terrestrial salamanders and their diversity relative to edge habitats and sampling type on a conserved property in northwestern Pennsylvania. So as I'm sure most of us know, there's a huge massive extinctions caused by human impacts right now, and amphibians are one of the most affected vertebrates of these declines. As you can see on this map, there's threatened amphibian species all over the globe, and these declines can be caused by habitat fragmentation and loss, disease like chytrid, and then invasive species too. But I'm going to focus on habitat fragmentation and loss because it's something that affects this area pretty strongly. And habitat fragmentation is when, say, like a road goes through a forest, and right along the road, the habitats are hotter and drier than in the middle of the forest just because of evaporation and the loss of the tree cover. And salamanders are a type of amphibian that are really affected by this fragmentation. They really prefer like living in streams and in damp woodlands. And it's super important to monitor their declines because they're keystone species, which means that they have a huge effect on the ecosystem. So they're predators on macroinvertebrates, and they also serve as prey in the leaf litter and in streams. So as I said, population monitoring is super important to understand these global amphibian declines. Monitoring can help inform conservation policy and land management. And also salamanders are great study systems because they have small territory sizes, they have high densities, low sampling costs, and they're an indicator species, which means they indicate microhabitat changes that are caused by habitat fragmentation. And local monitoring of species like this can allow for cheaper methods, quick results, and just fast action with the monitoring. But before you start a population monitoring program in an area, you want to test to see what sampling approaches are the most effective, because salamanders vary in life histories, and this can change in an area due to like the soil moisture or just like the type of salamanders in that area. So overall, natural cover and coverboard flipping are both effective for terrestrial salamanders, but natural cover flipping seems to be more effective, but it takes a lot more time and effort, and it also degrades the natural habitat. So you can see here me on the right, I'm flipping a log, and that just can mess up the decomposition that was happening underneath there by releasing it. And then the other picture is a salamander under a cover board. Another thing is that salamanders were used. In Meadville, Pennsylvania, we have a conservancy called French Creek Valley Conservancy, and they own property along the French Creek watershed. I think I have a map later on. Oh yeah. Oh no. Neighborhoods up top and the roads over up like Ilium Road and then the Conneaut Lake Road, and then the agricultural fields, the old ones in the middle. So it sort of allows me to look at like the fragmentation of the habitat. And also the Conservancy asked for me to look at this one, so that was part of my decision. So I set up 5 transects from Elion Road, which served as my fragmentation, to the forest core. So it's right along in there is where these transects take place. And I sampled from early April to October, and I took the last date from precipitation and the soil tiles, so a salamander to be under, so like a larger log or stick and a stone and stones of objects I flipped, and I obviously put them right back in their place, and then I recorded the time I spent looking in that box, and I identified the salamanders and just put them right back where they were. So I ended up finding only 9 salamanders total, even though I sampled from early April to late October every other week. So that was super surprising to me, only to find 9 total. And I found 7 redbacks and 2 red-spotted newts. And I found them mostly under artificial cover, so under that. I think at Mammoth Run, there was just, you would think to flip in the second box, so over here, just like once removed from the road, which was super surprising to me, and I don't really know why yet. So my preliminary recommendations, I haven't gone through all of my analysis yet, would be for the French Creek Valley Conservancy to sample in the fall, because I found no salamanders all spring and summer, and we did have a really dry summer in Meadville, but just to save their sampling time, just to constrain it to the fall. And that result is somewhat supported in the literature. A lot of people do sample in the spring and fall, but some studies, like the first one I listed, found more salamanders in the spring. So I think it just depends on the area you're in. I think in Meadville, and that's part of it. I think, I think it has mostly